Hello students, we are here with another video by Teaching Tube. Before we start, here is a gentle reminder. To stay up to date with my latest videos, don't forget to subscribe Teaching Tube. We have already learned a lot about cell and its organelles, but it's important to know how cell was discovered as well. So in today's topic, we will be learning about the device which was used to discover cell. Yes, we are going to look at two types of microscopes, as you need to understand how microscopy have developed and the differences between these two. So the first one we are going to study is the light microscope. It was first developed in the 17th century and it uses light as a source to form an image. And because of that, it can be used to view live objects. Its price is comparatively cheaper than that of an electron microscope. It can magnify up to 2000 times, which means that it can zoom up to 2000 times. So magnification is basically how much you can zoom into an object, whereas resolution is something else. Resolution is basically about the details. So it's all about the details you can see. Resolving power or resolution is the measure of the clarity of an image. It is the minimum distance at which two objects can be seen as separate objects. Like human naked eye can differentiate between 2 points which are at least 0.1 millimeter apart. This is known as the resolution of human eye. So it means that if you place two objects 0.05 millimeters apart, human eye would not be able to differentiate them as two separate objects. So the resolution of light microscope is very low, which is around 200 nanometer, and it is only found in one form. So now coming back to the other type, which is the electron microscope. It was first developed in 1930s and it uses electrons as a source to form an image, which means that it does so by firing the electrons through the specimen, which means it has to be dead. So it cannot, we cannot use it to view live objects. It has to be dead first, and it is very expensive as compared to the light microscope, as we already said. And its magnification power is a lot higher than that of the light microscope. So in order to know the difference between these two microscopes, light microscope and electron microscope, you need to remember the magnification power and resolution for each of these. Okay, and the resolution or resolving power for the electron microscope is very high, which is like 0.2 nanometers, and it is found in two types. One is SEM and the other one is TEM. Okay, so SEM stands for scanning electron microscope, whereas TEM stands for transmission electron microscope. So we will tell you the difference between what these two types are. So TEM, which is transmission electron microscope, is used to study the internal cell structures. Electron microscope has revolutionized the study of cells and its organelles, but it cannot be used to study life processes because the specimen must be held in a vacuum chamber. That is, all the air must be removed to study the life processes. For example, movement of amoeba, or to observe movement of amoeba, light microscope can be much better than that of the electron microscope. And in scanning electron microscope, which is the other type of electron microscope, electrons are reflected from the metal coated surfaces. So scanning electron microscope is used to study the structure of cell surfaces. The basic difference between these two is SEM is used to study the structure of cell surfaces, whereas transmission electron microscope is used to study the internal cell structure. So this one is for surface and this one is for the internal structures. Okay, so there are two types of electron microscopes. That's all about our today's topic. See you next time with another video on another topic. Till then, bye. Thanks for watching and don't forget to subscribe.